Today's talk is entitled Navigating the FDA's Expectations for Drug Approval, Adapted Designs for Dose Optimization and Accelerated Approval. My name is Kelsey Brown, and I am the Director of Medical Writing Solutions at TransPerfect Life Sciences in London, and it's my pleasure to be your moderator for today's session. This event is proudly hosted by the International Drug Development Institute, IDDI. IDDI is an expert center in regulatory statistics and clinical data science for pharmaceutical and biotechnology companies in several disease areas, including oncology, ophthalmology, CNS, and rare diseases. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI is headquartered near Brussels, Belgium, with a U.S. Center of Operations in Raleigh, North Carolina. Now it's my pleasure to introduce you to IDDI's clinical development strategy team and today's speakers. First, we have Dr. Elizabeth Elsecourt, Director of Statistical Consulting and Research at IDDI. ELSE directs the development, modification, and evaluation of processes for providing consultancy and study design support for clinical trials at IDDI. We also have Dr. Leonardo Garcia Barado, Senior Scientist at IDDI. Leonardo leads the statistical aspects of the design of experiments and methodological research projects at IDDI. And then we're also joined by Dr. Viva Deltuvati, sorry for that, Thomas and Dr. Vincent Staggs, who are both senior scientists at IDDI, leading a diverse range of projects involving study design, data analysis, modeling and inference, expert testimony, and regulatory guidance. So with that, I will hand it over to today's speakers.
Yeah, thank you, Kelsey. So welcome to our webinar on navigating FDA's expectations for drug approval. And let me maybe first talk to you about how difficult it can be to manage FDA's expectations. And I'm sorry, I have some issues with moving my slides. Yes, so if we want to keep up with FDA's regulations, we really have to watch carefully what is published. Like already last year, and these numbers are from the beginning of December 24, there were like more than 43 guidance documents issued by FDA. We have the CEDAR initiatives that are out there. We have pilot programs. We have FDA designations for making drugs available more rapidly. And then there's the different approval pathways that we will discuss, and the FDA patient-focused drug development, and much more. So to narrow down the content of today's webinar and make it less ambitious is, I want to say that we want to tackle this from a statistical perspective. We want to look at how innovative trial design can help with meeting FDA's expectations, and we will be focusing on Project Optimus, so on dose optimization, and on the accelerated approval pathway. So let me briefly introduce Project Optimus. That's a project from the Oncology Center of Excellence, and it became, I think, very known by everyone when last year this guidance document was published on optimizing the dosing of oncology treatments. And I think what the purpose is of this Project Optimus in a nutshell, it's really to reform the dose selection paradigm for oncology drugs. So where we used to have these cytotoxic chemotherapies that were development that are typically therapies with a short treatment duration, when the most severe toxicities are expected early, and only those were counted as DLTs. And importantly, it was safe to assume that a higher dosage would mean higher efficacy. So this means that the goal for these type of treatments was to define the MTD, the maximum tolerated dose, assuming that the highest possible dose would also have the highest possible efficacy. Now, for targeted therapies, this is different because these therapies, they are usually continued to be administered to the patients until toxicity becomes too high. So we also have to account for lower grade but more chronic type of toxicities, because those will determine how long patients can tolerate this drug. And importantly, we now no longer can assume that the higher the dose will just by definition gives us the higher efficacy. So that's why Project Optimus really stresses that the goal of this dose finding and this dose selection process should be to define the optimal biological dose. And that's the dose that's a trade-off between efficacy on the one hand and toxicity tolerability on the other hand. So let me dive into the implications of this Project Optimus. And first, I would like to talk about the implications for the early clinical development, so for the dose finding part. And as I said, the most important thing is that we have to move away from this maximum tolerated dose as being the objective. We should rather select a therapeutic dose range that will later be characterized in later clinical development, and we'll discuss that later. And this selection should be based not only on the toxicities, but also long-term tolerability, efficacy signals, and where possible, PK/PD data. And we will also, I think it's important that we integrate modeling and simulation with emergent clinical data. Now, the question arises, what is then the study design that we should use, that drug developers should use for the dose finding part? Clearly, it will no longer be the 3+3 designs that have been developed for these cytotoxic treatments to define the MTD, and we'll have to switch to a modeling approach. But there are so many modeling approaches out there. There are the model-based designs, model-assisted designs, and then should we model just toxicity, or can we model several outcomes at the same time? And then also, how will we select this therapeutic dose, so more than just one dose, for further evaluation? So, Vaiva, over to you to give us some further insights on these type of designs.
Yes, thank you, Els. So over to those finding designs. Obviously, just because of the time constraints, I will not have time to go into each design in depth, but I will really just go over the main families and discuss their advantages and the issues that we see with them. Hopefully at the end you will have, well, at least a broad idea of available options and will be able to make your choices. But while Els has just mentioned that the 3+3 design is a no-go, I will start from this design nevertheless, because it's still very popular. It's still kind of engraved in our minds as a go-to design. In this design, we treat patients in cohorts of 3, and the escalation, de-escalation decisions are made based on the target toxicity rates, which is usually chosen 30%. Which, given 3 patients per cohort, is easy to calculate at the back of an envelope. So no statistician is needed, rules are simple, and the design is easy to implement, which makes it extremely attractive. On the other hand, escalation is very slow. We might end up with many patients that end up treated in suboptimal doses. Another big drawback is that we use only the current dose level that is used to estimate the toxicity rates, which, given 3 to 6 patients, the estimates are very unreliable. So next slide. Given all this, an obvious improvement was to embed the information into a model which then allows sharing information across the dose levels. Continual reassessment method, which is on the next slide, is one of such model-based designs. In this design, what we do, we start by assuming some parametric form for the toxicity rates for those toxicity relationship. Sometimes we can go as far as including some prior beliefs in a Bayesian way. Then after each cohort, we update the probabilities of toxicity for all those levels. And this allows using all the information collected so far and leads to more precise estimates of the MTD. However, running models does require constant interaction between statisticians and clinicians, which sometimes creates operational difficulties and might, create some time lags between escalations and between moving to next cohort. So finally, some smart brains thought, well, why don't we just try to create kind of something that is sound based on some model, but that leads to a set of straightforward rules that can be easily predefined at the start of the trial and does not require any further involvement from a statistician. So these are model-assisted designs. And on the next slide, we have one of the most well-known of model-assisted designs, the Boyne design. It manages to get the best of both worlds. So statistically sound arguments. So we use some model behind the scenes to create the escalation, de-escalation rules. And so after everything is set up, the implementation is very straightforward. But the setup is more involved because we usually need simulations to investigate operating characteristics. But once the rules are in place, there is no more statisticians required. Well, the statisticians remain, but we don't need them anymore. So all that is nice and well, but as it was mentioned, these designs evolved based on the outdated notion of dose-response relationship that is parallel to the dose toxicity relationship, as you see illustrated here. This assumption was true 50 years ago when most of the drugs that have been used in oncology were cytotoxic. But over the past 10, 20 years, we've been moving away from these towards more targeted approaches. And these novel drugs frequently have much wider therapeutic index, show much less toxicities in therapeutic doses. And if we plot those response and those toxicity relationships next to each other, we might see what you see on the screen right now. So rather than the expected parallel profiles that we assume for the designs I have just presented before. So this is why we're moving from maximum tolerated dose as means to select the dose for further development, but rather the efforts are now focused towards finding the optimal biological dose. And this dose is defined as the one that has tolerable levels of toxicity but maximizes the efficacy. The designs that are that I will just quickly present afterwards are just stemming kind of an extension from previous designs, but we introduce another dimension. So for example, the FTOX design. Hello, can we move to the next slide? Yeah. Did you lose me for a long time?
Just a few moments.
Yeah,
I think I was
go on, Viva.
Yeah.
It was interesting.
Okay. So I'm going on with the talks. I'm going for a talks design, which is based on assuming two models for dose toxicity, one for dose toxicity and one for dose efficacy. And based on these two models, we can draw the plot that you see on your left side. And depending on where we want to fall, you know, how much toxicity we want to allow and how much efficacy, we can draw the so-called utility contour. And so again, based because this design requires statistical involvement, is more involved. It does require closer cooperation between statisticians and the clinicians. Another design that is also using the idea of utility is the Boyne 1-2 design, except that utility here is much more simple. We just assign weights, and based on these weights and the observed toxicity rates and efficacy rates, we can quite easily tabulate the decision rules, and so this design is again model assisted design, so we have it more involved behind the scenes, but once everything is set up, it's easy and straightforward. Boyne ET design doesn't use the utility function, but it's based on toxicity and efficacy rates observed at the time of the decision making, and so the decision rules again can be pre-specified ahead of time. So now we have these two-dimensional designs, and they do give a pretty good idea about where the optimal biological dose could be. But the choice would be incomplete if we really skipped on other essential aspects of the drug, and such as pharmacokinetics, pharmacodynamics, pharmacogenomics, and the long-term safety. So, so far there are no models that can do everything, but what we can is at least take them into account when making decisions. So here's just the quick summary of the designs I have just presented. So, just, you know, as a recap. And I think we can move on with the presentation now.
Okay, thank you, Viva. So we just talked about the implications of Project Optimus in the early clinical development, so really on the designs that we use for the dose escalation and the first dose finding. So let's now move on and let's talk about what the implications are of Project Optimus for the dose optimization in the later clinical development. And so what I think is an important implication is the pre-approval requirement, is that so in this new guidance document, FDA clearly lined out that we now have to do the dose optimization prior to drug approval, and it will no longer be allowed to have first a drug approval and then have as a post-approval requirement some further dose optimization trials. The other implication that I think caused the most consternation when the guidance document was out is that now FDA asks for a randomized comparison between different dosages, and this has become essentially mandatory. And another consequence is that the drug developers are asked to incorporate safety information that goes beyond DLTs, so also includes tolerability. When I said that the need for randomization, it caused consternation, I don't think it's because drug developers are not in favor of the principle of randomization. We all know that this is crucial, because if we, in a typical dose finding trial, we would look at the different doses in different cohorts, and because it's not randomized, we can't be sure that the differences that we see, that those can be attributed actually to the different dosages, but it might also be just by the play of chance that we have differences in the cohorts. If we bring in this randomized comparison of a few pre-selected doses, this will allow us to further characterize the different doses for many different aspects in an unbiased way. But what caused the consternation, of course, was that it sounds like when you have to do a randomized comparison of doses, that this could increase even further your cost and time that is needed for the dose selection process. And it's already so costly, and it's already such a long process. But I think here, as statisticians and methodologists, we can really say that, well, it really depends on how you implement this dose selection process. There are some aspects of this process that we see are often done in current practices, like using expansion cohorts for different doses, but in a non-randomized way. I don't think we should do too many of those. Maybe we can think when we do backfilling of doses, so when a dose is cleared and is considered safe for further evaluation, we could wait until more doses are cleared and then randomize this backfilling or a further enrollment of patients to these doses. And it's also important to note that in the guidance document, it is clearly said that by no means it's needed to power this dose optimization trial for dose comparison. That would lead to prohibitively large sample sizes, and that's not needed. So I think here we have to use innovative trial designs and think careful about the designs that we use. When we think about innovative trial designs, of course, we talk to many pharma and biotech companies, and those trial sponsors are also very innovative when it comes to questions they have and what types of designs can we use. So questions we often hear and we often think about are, for instance, can this randomized dose optimization part, can this be included in a seamless phase 2/3 trial? And that then opens new sub-questions. It leads us to other questions like how to size then this dose optimization phase and how to decide on the dose selection if we don't need to power for this dose comparison. And what type of efficacy endpoint would we use? And thinking about this efficacy endpoint for dose selection, that often is an earlier type of endpoint and not the endpoint that will be used to evaluate clinical benefit. The question comes up like, but can then this trial combine objectives of dose optimization and approval? If we think about the accelerated approval, that is typically done based on also an early efficacy endpoint. Maybe one slide on this accelerated approval pathway. I think many drug developers will be familiar with this. It is a pathway that has the objective to get drugs for serious conditions, filling an unmet medical need, to get them faster out there for the patients to have access to those. And this is based on a surrogate endpoint or an intermediate clinical endpoint that is typically measured earlier than the endpoint that is used to establish clinical benefit. And then after this accelerated approval based on the early endpoint, we see that there is another trial or the trial continues to evaluate this confirmatory endpoint. Now, in a draft guidance on accelerated approval that FDA published in 23, the changes there was, well, for me, that was, I think the main change was that FDA required that the confirmatory trial is expected to be fully enrolled or close to fully enrolled before a sponsor can start discussions on accelerated approval. And the guidance document mentions two strategies for this confirmatory trial. So the first is the classical one. It's a phase 2 trial based on this early endpoint, and then followed by a confirmatory phase 3 trial. But they also clearly mention now this one trial approach, also referring to a seamless phase 2/3 trial, in which you would enroll patients, follow the patients up until you can do an analysis of the early endpoint that will be the basis of the accelerated approval, and then enroll more patients or follow up the patients for a longer period of time to then evaluate the confirmatory endpoint or the endpoint that will show the clinical benefit. And just for the sake of completeness, there's also, I think, in December 24, late last year, FDA also issued this guidance document on accelerated approval outside of oncology therapeutics. But so you see, there's a lot of discussion, I think, in the drug development community about trial design and about the methodology. Like, there are so many options, but what can we combine into one trial? And is this one methodologically feasible? And is this also beneficial? So, Leandro, over to you to tell us something more about these type of trial designs.
Yes, thank you, Els. So I will discuss here somewhat, or try to go into somewhat more of detail, or whether like this one trial that includes all the elements that we just have discussed, whether that's reality or still fantasy. I will focus mostly on whether it's feasible not as much as on how useful it could be, because that's a kind of a more difficult question, or whether you actually should use this kind of methodology. So basically the question where we will start is whether this is seamless dose optimization / pivotal trial, so whether we can combine these two, whether that's possible. So on the next slide, our starting point will be we have successfully performed the dose finding trial. We have, in doing so, incorporated all available information at the time, and we have selected a set of doses to consider in a dose optimization trial. So the question now is like, what should we do next? Like in what way could we actually select the optimal dose? How would it fit into, for example, a pivotal trial, and are there any shortcuts so that we can actually make this happen? So what I look at is on this graph that you see here. First option would be like an operationally seamless design. So basically this is like somewhere between two completely separate trials, or a trial that would seamlessly go into each other. So you see here the black dashed line that actually makes a hard connection between the two trials that we consider here, so the dose optimization and the pivotal trial. So basically you plan them to be quite close to each other, but you still consider them as two different trials. So this has advantages that is straightforward to do your type 1 error control. There's no need for a control group in the optimization trial. And it allows for some time between the trials, which means that you can have a fairly good idea about longer term toxicities or tolerability if you consider this time between the trials. Disadvantages are there's no way to include the information that you already gathered on those two, for example, here into the optimization trial. There's no calibration information in your dose optimization part, which means that what you're seeing for the doses, depending on, let's say, temporal effects, is perhaps not that different from what you would see in a control, but you don't know because you don't have it. And also a disadvantage is, of course, that there's some time between the trials, making the overall time duration a bit longer. We could already think about basically connecting these two phases together. So we now see that there's no stopping between the trials, but for example, those two information will be collected during the dose optimization and will just continue during the pivotal. And basically we introduce here an interim analysis to make a decision about which dose we would carry forward to the pivotal trial. At this point, advantages are that there is available calibration information. We can include the dose 2 information from the dose optimization into the pivotal trial, which kind of makes probably the combined trial duration shorter than what we were looking at before. In terms of disadvantages, now there's a control group required in optimization, because if you want to carry the information forward for one of the doses, you also need to have some information about the control arm to be carried over. And there's reduced time between the trials, so there's somewhat less options here, or it's a bit more difficult to look at longer term toxicities and tolerability if your overall trials are getting shorter. But the most important part that we have to take into account here is that there needs to be some additional type 1 error control, because you do use information from the dose optimization or you carry it over to the pivotal, so you need to take that into account. So how do we take this into account? So how can we have these different decision options that actually can control for the fact that our type 1 error may be increasing? So one option I would consider, I call the alpha splitting, is basically just consider a formal test, which means that as stated on this slide that you see here, you basically need to account for the fact that you have multiple comparisons, because now in this dose optimization part, at this interim analysis, you will have a comparison from each dose to a control. So you need to kind of correct the alpha, the type 1 error that is potentially happening there. You have to correct for that. This you can do basically with the usual suspects. So you can actually split your alpha over all these dose comparisons over the different doses by using, for example, just Bonferroni correction. In addition, you also test now, or you have multiple testing in every dose arm, because you have a test at the interim analysis, but as well as at the final analysis. And here, for example, you can correct for this by considering hierarchical testing, which means that within, for example, these dose 2, you will only you can, let's say, carry over the considered alpha level that's left to the final analysis, because there you have kind of already made this decision. So if you get to your threshold, you can actually carry over the alpha level that you have there. In the pivotal trial, you can actually use the allocated alpha, so the one that you would have left in every dose arm, or in the dose arm that you have taking over to your liking. So you can just do a fixed sample size design, or a group sequential design, or an adaptive design with the usual methodology. This kind of brings us to a second possibility of type 1 error correction, which would be like what I would call the group sequential design. And in this setting, we can actually consider the method that was proposed by Stelz and Todd in 2003, which kind of makes use of, let's say, score statistics at this dose optimization interim. So what you would basically do here is you calculate score statistics for every dose, and you basically pick the one with the maximum value, and that will be the one that you will graduate to the pivotal trial. In addition, this allows for early stopping. You can even choose, let's say, the dose that did not provide the largest value, so that did not have the maximum score statistic value. Type 1 error control will still be okay, but you will kind of lose power overall in your trial to do that. So that's also a possibility. And the way that type 1 error control is actually controlled in this way is that this method involves calculating the critical values for every subsequent decision, so for the final analysis or other intermediate interim analysis, which are based on the theoretical joint distribution of the test statistics. So basically you control for the fact that they're correlated because you defined that in such a way. And because you use this theoretical framework, it means that if the assumptions of this theoretical framework are satisfied, these are mainly normality assumptions, it will be very efficient. It also allows for additional interim analysis after the dose selection. Somewhere between an advantage and a disadvantage is that it's very good in selecting one of the best. So if you have in your dose set, let's say a subset which is well, very good, or is just better than the rest of the doses, it's very good in selecting one of these. If you really are going to the best of the best, probably a different method will be more suited for that need. And perhaps a disadvantage, depending on what you want in a particular trial, is that all interim analysis in this case have to be pre-specified. Otherwise, you cannot embed it in this theoretical framework. Which then brings us to a third option that I will consider here, and that's the case when you don't want to have everything pre-specified. You can actually go to a general adaptive design. So for example, you can imagine that, again, we will be looking at an interim analysis after our dose optimization. But now we don't, or we see the data, we don't decide that we will take one dose to the pivotal trial, but we are kind of postponing the selection and want to take up dose 1 and 2. Before at an additional interim analysis, we actually make the choice of choosing dose 2 to go to the pivotal trial. And even at that point, we may decide on an additional interim analysis. So basically, under this methodology, you can have unplanned adaptations where it kind of invokes the yeah, so we can go back to this slide. So you have this closed testing principle where you can actually account for the way that you have these multiple comparisons, which means that you will have these adjusted stage-wise p-values, and the decisions you have at this multiple stages, which upfront you don't know how many there are. If you want to do this unplanned, you can actually combine them into with p-value combination tests, so that you have full alpha control over all the decisions and results that you will have in this trial. So another interesting idea here would be, now let's say that we want our decisions to be based on early endpoints. So what we've discussed before all assumes that the endpoint we have at our interim analysis is the same endpoint as we will be looking at the final analysis. Now we can say, let's say that we do the dose optimization, but we pick our dose based on how they perform on an earlier endpoint. And then still we carry forward with that dose, but then at the end of this final analysis, we will use a different endpoint, which will be our final endpoint that we will use there. Again, well, Todd and Stellar updated their 2003 paper or their methodology, and actually came up with a new method for this, or extended their method to account for this, where you see that the slide is exactly the same except for the final bullet, which is here in bold, where if you, on top of what we've discussed before, now assume a correlation between the test statistics based on this early and final endpoint, you can actually still use this updated method to actually incorporate them. And then this correlation, depending on what the value actually is, can have an impact on your operational characteristics, but you can actually calculate what this would mean in terms of what you can expect for such a trial. This, of course, leads us also to what we consider this accelerated approval pathway, where you can think now that, well, you have a set of options here as well. So one example of such a trial could be this one, where you have an interim analysis where you decide on which dose to carry forward based on this early endpoint. You start your pivotal trial, and then at some point you have an additional interim analysis, again on an early endpoint, where you can actually try to file or have some information for a potential accelerated approval. And then that would continue on, because if you get approved, then you can use a final endpoint to actually state that this was actually the case. So this is an example of this single trial accelerated approval. Again, you can account for the multiple testing because you have this accelerated approval interim analysis and this final analysis in the pivotal trial. You could consider here this multiple testing, or you can consider the alpha control level here by assuming hierarchical testing, so all in, which is what you could assume in this general accelerated approval framework, where you would only proceed when you have a significant interim analysis. So this kind of makes sense in some cases. In other cases where it's not crucial to also show, let's say, a significant treatment effect on this early endpoint, you can consider a fallback procedure where you would reserve some kind of type 1 error probability to spend at the final in case you would not reach a significant result at this accelerated approval interim analysis. So the trial can continue. So basically, as a concluding remarks, I could say that indeed, I mean, these type of trials that you can envision, which actually take everything into account, where you can also have these ideas about accelerated approval testing, can actually be considered. Why, or if you should consider them, that's of course another wheel. So I would say there's no need to reinvent the wheel. We have shown that already a lot of things can happen methodologically. So these are exciting times for us because it's very cool to see what like of the old methods could still be used, whether they could be improved, and how they actually would fit into a particular trial. In case of the seamless optimization pivotal trial, there is always a trade-off, so we have to be very careful with that and have to monitor that for a particular case, because there is a correction happening in terms of the dependence between the test statistics, and that correction can actually increase the sample size. You have to make sure that you actually are getting to a stage where you still have what you want. And this gets worse when considering this early endpoint, because then an additional correlation gets into play. In terms of accelerated approval, I think it's important to make it operationally sensible. So you need to have enough information at the optimal dose selection. If you would like to do, let's say, the accelerated approval interim analysis, you would like to coincide that with the actually the dose selection, and you would need to have enough information at that time. And that may be difficult because there is a regulatory requirement that says that you would almost need finalized accrual at the moment that you would file for the accelerated approval. So that could make your optimum or the dose selection or the dose optimization part longer than you would actually want it. So it's a good thing to be mindful about that. Seamless all the way, like integration of all information may not be that straightforward, so maybe additional methodology may be needed there. There's also no one-size-fits-all solution, so the most efficient or optimal design, it's really important that you define that on a case-by-case basis so that you don't have too much correction for type 1 error control, for example, that you actually blow up the sample size. I think as a final statement, something that we all strive to do is don't make things more complex than needed. So it's nice to have this methodology, but we should use it with care. And with that, I would say over back to you, Els.
Thank you, Leandro. So thank you also for this introduction of group sequential designs with its extensions. And I think the group sequential designs, it's a type of design that we are all used to work with in drug development, but these are really such flexible type of designs. They introduce interim analyses that allow us to stop early for efficacy or for futility. And so in the end, it can really reduce the expected sample size of the trial. Now, I think here this is a type of design that is especially of interest when a large trial is designed with limited prior information, and that's typically the case for the seamless phase 2/3 trial, so that we can stop this trial earlier if the treatment effect is larger than we anticipated, but continue until the end, that we have kind of a safeguard we can continue till the end if this is needed. So we know that these group sequential designs, as Leandro also said, it's a well-known theoretical framework, and these are very efficient designs. So it means that for a given sample size, we have a good power to detect a certain treatment effect. Now, the way they are set up is that they are powered for a conservative treatment effect, so for a smaller effect, meaning that the total sample size that might appear very large, but we know that the expected sample size might be smaller in case we have underestimated the treatment effect. But there is another type of adaptive design, the sample size re-estimation trials, that works a bit the other way around. So the sample size re-estimation would power a trial for an optimistic treatment effect. That means that the trial would have a smaller initial sample size, what many drug developers would like, but we do commit in these type of trials to increasing the sample size if needed, but whether it's needed, that will be based on the interim treatment effect. Now, the question is, well, especially I think for many statisticians, is that we know that group sequential designs are so efficient. Are these sample size re-estimation approaches also efficient? And when would those be the better design? And then also in terms of methodology, there are so many different sample size re-estimation methods already published. And so what would be a good method in which case? So please, Vince, over to you to tell us something more about this.
Okay, thank you, Els. Am I able to advance the slides? Oh, you've got it. Okay, great.
Yeah.
So just recap just a little bit from what Leandro and Els have talked about. We can define an adaptive design in general as a design where that final sample size is not predetermined, but rather how many events or how many participants we end up with is a function of some look at the data after the trial has begun. So with the sample size reassessment, the final sample size is based on an interim analysis, just like with the group sequential design. But here the idea is we run this interim, and then based on the conditional power at that interim analysis, we can increase the sample size further going forward to ensure that we have the power in the range that we want or that we've targeted. And there are a couple of versions of this. Typically, these are unblinded interims. So at the interim, we have to have someone who's unblinded estimate the treatment effect, so we can estimate the conditional power and then make the adjustment to the sample size if needed. But you can also have sample size reassessments where you're not focused on the treatment effect or effect size, but you have some kind of nuisance parameter that is going to affect the variance. And initially, you may not have a good sense of what the value of that nuisance parameter is. And so you can set up a sample size reassessment where you take an interim look at the data, but you remain blinded. So you just look at the data as a whole. You're not looking at the arms separately to try to get a better estimate of that nuisance parameter. We had a client we were using a negative binomial model, and you of course have this over-dispersion parameter. And so we set up a design like this so that we could take a look at an interim just to get a better estimate of that nuisance parameter, but not actually looking at the effect size. We can go to the next slide. Thank you. It feels like, I think, to many people that if you're going to take an early look at the data, and here I'm talking about an unblinded look, and look at that effect size and then change the sample size, mid-course, it feels like maybe we're cheating a little bit, right? Because we're used to sort of fixing the sample size and then running a study and dealing with the p-value that we get. So there is this question of type 1 error rate inflation, and when does that happen if you do a sample size reassessment? So you can show theoretically that when you run the interim, if the conditional power is greater than 50%, there's actually no need for an adjustment to your alpha level. You can do the sample size adjustment, increase it as needed, as planned, based on that conditional power, and you won't inflate the type 1 error rate. And in fact, it could be lower than 50%, and you can read a paper about this and try to figure out exactly what that is. But assuming you need to make an adjustment, there are various adjustments available, and they're not too difficult to implement. Next slide, please. So we focus on promising zone designs, and the idea here is that if you're going to do a sample size reassessment, you're going to increase the final sample size if and only if the interim test statistic falls in a promising range. And so what we mean by promising is that it's sort of not too hot, not too cold, but just right, like Goldilocks. So if your interim sample size or interim test statistics looks great, and you already have, say, 90% conditional power, we don't need to increase the final sample size. We'll just let the trial run to the planned sample size and stop. And if the test statistic interim looks really bad, we're not going to want to invest more sample size in the study. So we'll just, again, not going to increase the sample size. We'll just let it run, the study run until we hit the planned sample size. So promising is in between these. It's not unfavorable or disappointing. It's not favorable, but it's promising, right? And so the idea is here we've got enough signal that there's a meaningful effect, that if we increase the sample size, we can get conditional power up into our target range. And so that's what we'll do. Next slide, please. You can see in this figure depiction of the promising zone in terms of the Z statistic value at the interim. And the Z statistic is on the horizontal axis here. On the vertical axis, we have the total sample size. So that could be events, if it's a time to event analysis, or it could be number of participants, if it's not time to event. On the left side of the graph, you can see the total sample size is going to be about 280 here, unless we see a Z statistic at the interim in that promising zone. And if it's in that promising zone, we're going to bump the sample size up to about 420 in that first, between the first two dotted red lines, dashed red lines. And that's the max that we've set ahead of time, is for 420. So we're going to go ahead and go up to the max to get power as high as we can. And then you can see between the second and third dashed red lines, there we're going to increase the sample size all the way up to the point where we hit our highest value of power in the target range. So I think this is set up for a target range of 80 to 90% power. So the idea is we increase either up to the maximum sample size allowed to get power as high as we can get it, but we don't increase to go over 90% power. So in those cases, we just go up to the point where conditional power hits 90%. And then off on the right, the rightmost segment of the graph here, again, there's no increase in sample size there because the Z statistic interim is so large and so promising that we're just going to stick with the original planned 280 or so participants or events. We can go to the next slide. Oh, thank you. You're ahead of me. Great. In practice, there are some nuances to these designs. Sometimes on the actual test statistic scale, what we find, and when we implement these, we run simulations. So we simulate the whole data generating process, and then we get the operating characteristics. So we're interested in power, obviously, but also we look at how often in our simulations the interim actually triggers a sample size increase. And it's surprising sometimes because that sometimes it doesn't happen very often, right? And maybe you're only going to increase sample size about a quarter of the time. There's also an interesting, you can show this theoretically, an interesting finding that because when you see an unpromising result at the interim, we don't actually increase the sample size. You're not actually maximizing power with this constrained promising zone design. If you really wanted to maximize power, you can kind of think about it like when you see these sort of disappointing results at the interim, that's when you really most want to add more events or more participants, right, to get the maximum power, because that's sort of where you need to help the most, right, is for that disappointing result. We can go to the next slide.
Yeah. So
Back
I
to you,
think,
Els.
yeah, thank you. I think we've come to the end of this webinar, and I will just quickly summarize. So I think it's fair to say that Project Optimus, it has many implications for dose selection. So we discussed this moving from the MTD to the optimal biological dose, that we are expected to have a randomized comparison between different doses, that the dose selection should be based on a variety of information, and importantly, that all this is now a pre-approval requirement. But at the same time, I think the Project Optimus also has many opportunities for dose selection. And as Leandro said, these are exciting times for methodologists and statisticians. So we should focus more on modeling all of the available clinical data. And we have the opportunity to think through all the different approaches and find the one that is the best for a given indication and a given product. We could even think about including the dose optimization in this one trial approach for accelerated approval. So I think for us, the trial design is key to make the most out of this new paradigm for dose optimization. So with this, we've come to the end of our webinar, and I hope you enjoyed it. So in case you want to know more, you can go to our website. We have other resources posted there. We have webinars, other webinars, but we also have podcasts on accelerated approval and on Project Optimus, diving a bit deeper into those topics. And in case you have questions, please send them over to us through our website and we'll be sure to return to you. Thank you.
Thanks, everyone.